So we will have Anders introduce himself. Would you like to stand and wave?
Yep.
We will use the blackboard later. So we'll show him at the blackboard now. Let him introduce himself, and then if you want, he'll switch to the slides, and then he'll let you know when he goes back to the blackboard, to the whiteboard.
We ready to go then? Okay. Hello. My name is Anders Carlsson, and I have a very forceful personality. Now, by that, I don't mean that I push people around, but I spend a lot of time thinking about force, and especially force in cells. And to me, that is the essence of mechanobiology, understanding how forces are generated, controlled, and measured in cells. So that's going to be the general topic of the talk today. And let me encourage you to ask questions whenever they come up. You might need to shout, but please go ahead and do that. And as Patience mentioned, I'm going to do kind of an Ant-Man thing where I'm going to shrink. You'll be shrinking me to a smaller screen or expanding me to a larger screen. So are you running that, Jim? Besides me? Okay, great. Then I'll just tell you when I want to do the board or when I want to do the screen. Okay. So let's look at the screen now a little bit. There are several different types of forces in cells. One that I've been interested in over the years is forces from polymerization of biopolymers. I'm getting a bit of an echo. Okay, and so we'll look at those. Those are rather small scale forces on the scale of individual filaments or a few filaments. And then we'll also look at forces from whole cells and collections of cells. And then we'll look at other types of forces, including osmotic forces and also the kinds of forces that open channels in cell membranes. But the first thing is we need to decide what is force. And in this case, it's not as obvious as it seems. So let's have me at the blackboard now. Expand me. The same stuff is on the little screen. So if you can move forward one slide, Patience. It's on there, but I'd rather do it on the board. Imagine now that you have a cell on a substrate, and you have a number of different forces acting on the cell. Then what our first year mechanics in physics tells us is that the total force is equal to mass times acceleration. So this is how we look at, for example, weights falling in the force of gravity. But it turns out that this is not always that useful in biology. And the reason is that often the forces add up to being very close to zero. So for example, let's think about an accelerating cell. So we'll say that F total is mass times acceleration. And the mass of a cell is like volume of the cell times its density, which is about the density of water. So that's 1 gram per centimeter cubed. And then times, what's the acceleration of a cell? A cell might accelerate at a very small rate. It's moving at like a rate of some hundredths of a micron per second. So the acceleration might be 100th of a micron per second squared. And finally, we would take that times the acceleration times the So that would be all the pieces. And when you finally work this out, this comes out to about 10 to the minus 8 peak newtons, which is the same as 10 to the minus 20 newtons. Okay? So this is actually a very small net force. So in fact, there's basically zero force acting on the cell as it moves. And in fact, you know from your mechanics that to keep an object moving at a constant velocity requires no force. So there's really very little net force on the cell. And the cell, as it moves through a viscous environment. But it turns out that from that, it's very small as well. Yes, was there a question? So in general, the total force on biological. Is somebody
Echo on it. Okay.
okay. Let me know when I should start. Should I start? Okay, very good. So the bottom line is that we don't really have much of a net force. Instead, we have a balance of forces. And think of this, here it is, leaning on the board. And actually on it relatively quickly. There are many kinds of forces, but there's no motion. And this is typical biology, that there is balance of forces between the components. And that's what we're exploring further. Okay, back to the screen now. You can minimize me, and let's go back to the PowerPoint. And let's move on to the next slide. Okay, so this is a familiar example of the balance of forces. You see that there's a rope in the middle, and two people are pulling on this with their hands. And there's no motion, there's no net force on anything really, because the forces are always balanced. And we describe this as being a tensile stress, or saying that the string is under tension. And that's often the situation in different parts of biological cells, that they're under tension, but nothing is actually moving. And there's another kind of stress that occurs, and that's called shear stress. If you look at the bottom picture, you'll see that there is a block sliding over another one. So let me get the pointer on that. Yep. There. Okay. So if you look here, these blocks are sliding relative to each other, and that sliding is resisted by both of them. They don't want to change their shape. And so that kind of sideways stress is called a shearing stress. Whereas on the other, in this situation, where the forces are perpendicular to that surface between the objects, this is called a tensile stress. And if it were pushing in, it would be a compressive stress. So, next slide. Okay, and here's some examples of how forces balance out in cells. On the left-hand side, you see that there is an internal pressure from the inside of the cell. And do any of the students there know where that pressure comes from? Okay, any ideas where that pressure might come from? No? Okay, well, the fact is that there is, in most cells, there is an osmotic pressure coming from solutes inside the cell that pushes out on the cell membrane. So that is these blue arrows that are pushing out on the membrane. And that would tend to make the cell expand. But there are other forces that contract the cell, In fact, there is a cortex around the edge of the cell, which tends to expand and pull it in. And there is also a tension in the membrane of the cell. And these factors then balance out so that the cell has a certain equilibrium volume. And if you'll go to the middle here, there are, again, a couple of competing forces. One is that the cell would like to stretch because it likes to attach to the substrate. There is, the red bars here indicate a binding between the cell and the substrate. But on the other hand, in order to do that, it has to stretch the cell membrane. So these two forces are fighting against each other and balance out to an equilibrium configuration. And something very similar here is happening on the right. Here, the cells would like to attach to each other. They're binding to each other. And so that makes the boundary want to expand. But on the other hand, that changes the area of each of the cells. And so there are opposing forces. Okay. Yes? Well, thank you. That's a good question. In this case, they're actually balancing out. So the cell has reached a configuration where the forces from the membrane are equal to the adhesive forces. So we're looking at those components. So there's actually a rather precise balance between those. But I should say that apart from just the membrane itself, may be a lot smaller. What is making the cell, what's preventing the cell from getting as large as it wants is first the tension in the membrane itself, but then there's also an active force from the cortex, and that active force is often larger than just the force from the membrane. So I think the answer to your question is that just the force from the membrane is, in most cases, smaller than the force from the adhesions. Is that what you had in mind? Let's see. Right. So the question is, when a force is applied, is it more likely to rupture the cell adhesions or to rupture the membrane? Well, let's see. There are cases, for example, one case where the membrane gets ruptured is if the internal pressure gets in the cell gets too high. But I believe in that case that the adhesions would release first. But that would depend a lot on the state of signaling of the adhesions. If the adhesions are very tight, then I believe there would be enough force there that the membrane could actually get ruptured. Let's move on. Okay, so this is the we want to get quantitative, and we often talk about energy in units of the thermal energy. So this is the thermal energy, and that means thermal energy means that if you have a molecule in a gas at a certain temperature, that its kinetic energy will typically be this number, which is about 4 piconewtons times nanometers, or about 4 times 10 to the minus 21 newtons times meters, or joules. And later, when we talk about pressure, we'll be talking in units of megapascals, and the megapascal is about 10 atmospheres, so that's a rather high unit of pressure. The pressure in a car tire or a bike tire is a few atmospheres, so a megapascal is a high pressure. Okay, so now how do we actually measure anything about the mechanical properties of a biological system? We need a reference scale in order to get a handle on these forces and energies. And the thermal energy actually can provide a useful reference scale. And in fact, if you look at the thermal fluctuations of an object, that can actually tell you how stiff the object is. Because a very floppy object will have larger thermal fluctuations, and a stiff object will have much smaller fluctuations. So can you advance one? So now we have a movie. Here, this is a couple of filaments of the biopolymer actin, and they're overlapping. So along their central part, you may be able to see that it's a little brighter. That's where they're bundled. But the idea of this experiment is that this filament is simply fluctuating thermally. The physics analysis tells us that there is a relationship between these. Can you go back and forth? Between these fluctuations and the stiffness of the filament. And we measure stiffness of filaments in terms of something called the persistence length. The persistence length is really the typical radius of curvature of the filament. So if the filament is very stiff, it's very straight, then the radius of curvature is going to be very large. And here you can see that it's bent, you can roughly make out the radius of curvature in this movie. If you look, for example, at this end, you can see it bending with a certain radius every so often. Sometimes it goes completely flat, other times it bends. But from looking at these kinds of movies, we can figure out what the persistence length is, and thereby quantify how stiff the filament is. And so on the right here, I've given you numbers for some typical biopolymers. So DNA is actually very floppy. Its persistence length is only 75 nanometers. Actin is stiffer. Its persistence length is 10 microns. Actin filaments, on the other hand, in cells are typically much shorter than that. So they're basically straight and they're bent a little bit. Microtubules have a whopping persistence length of 2 microns, or 2 millimeters, excuse me. And can anybody guess what the persistence length of a piece of spaghetti would be? Imagine you had a very, very long piece of spaghetti. Okay? Anybody care to hazard a guess for the persistence length of a piece of spaghetti? Is the mic on there? Kilometers? What's that? Oh, thank you. Yes. Thanks a lot. I've only done the measurement for the old cooked one. Uncooked. Yep. 3 meters. Oh, okay, 3 meters. Any other guesses? Okay, 100. Okay. Right. All right. Good. Well, let's look at the number here. There we go. It's actually 100 light years. Okay. So that would mean that if you had a piece of spaghetti and it had no forces acting on it, it only had the thermal energy, that then it would be incredibly straight. And now, of course, if you think even of an uncooked spaghetti, it's not all that straight. You're holding it, it bends a little bit. But whenever it's bending because there are active forces. You might be holding it, but then gravity's pulling down on it. So this is just a way of telling you how small the thermal energy actually is. It would only bend the spaghetti by a very small amount. Okay, so now let's talk about polymerization forces and how they're generated. So what is polymerization? Well, here's a polymer. This is it's supposed to be a model of actin, but it's a mathematical model that I made, so it's very oversimplified. But nevertheless, it's a lot of individual subunits. So here is one protein that's like 5,000 atoms. Here's another 5,000 atoms, another 5,000 atoms, and these proteins are forming a filament. And at the end, the filament can grow. And because the growth is exothermic, it's energetically favored, and it's energetically favored in such a way that it can actually overcome an opposing force on the membrane. Okay, so here's a cartoon of how this happens. I think if you can just step forward one, that should do it, or maybe I think I can do it here. There we go, thank you, yeah. Okay, so now here it's happening, this filament is wiggling and fluctuating, and there's a monomer that's gonna come in at the end, and you see that it attaches to the end of the filament. And it's actually possible to generate force by doing this. So you see the membrane there in blue, it's moving with thermal fluctuations. But there is some force against it. Maybe there is something that's stretching the membrane, kind of pulling it back against the filament. But the filament can overcome those forces and then make the membrane protrude. And so one thing that we're interested in is how does the growth velocity depend on the opposing force? And that's something a lot of people have calculated. Here is the calculation that I did some years back. And what you see is that the force scale on the right, it's like this is actually roughly in piconewtons, and this is the scale velocity on the left. So you see that by the time you get down to a few piconewtons, it's kind of pooped out. Okay, so the theory says that you should be able to generate forces of a few piconewtons with this kind of growth mechanism. Okay, now go to the next one, and Jim, can you maximize me again? Okay, so this is a very clever measurement of the polymerization force of a single actin filament. It's actually very hard to measure this force because actin filaments, if you let them get longer than a micron, they become actually very floppy and they can't generate any force. And that's because they buckle. And what I'm talking about is this. So imagine that this is your actin filament. You've got a little hand that's holding it here and another hand here. And then you push down on it. At first nothing happens, but then all of a sudden it buckles, and it buckles randomly. It could instead go in that direction. Okay, but a filament will buckle eventually if you compress it. And here in this ingenious experiment, this is being used to measure the polymerization force. So Jim, please minimize me now, and then we'll go back to the screen. Okay, so this experiment, there is a filament of actin that's bound to a substrate with different proteins here and there. It's growing at one end. New subunits are coming in at this end, and at this end it's just passively attached. And remarkably, at this end, the new subunits can add, even though it's attached to the substrate. Okay, so let's see what this does. Okay, there you see it grows and it buckles and then it turns out. Let's watch that one more time. See, at some point it buckles, and then eventually it wasn't interesting after a while. But the reason this is useful is that we know what the buckling force of an actin filament is. And it depends on the length. So our formula here is it's various factors. It's the KT that we have from before. Here's the persistence length. And the L here is the length of the filament. And for the length here, that works out to 0.8 piconewtons. So this tells us that a single growing actin filament was able to generate a force of at least 0.8 piconewtons. And that's actually one of the best numbers we have for this particular kind of polymerization. But for microtubules, we can do better experiments. Microtubules are another kind of biopolymer. They're, as we saw before, they're stiffer than actin filaments. And so you can have a relatively long and visible microtubule, and that you're still able to measure its properties. So in this experiment, if you look here from the side, this is a slide with a well in it, and there's a microtubule lying on the substrate and pointing in here toward the edge of the well. And if you look at this in a top view, take a look at this filament here, you'll see that it grows and then at some point it bends, and that's because it hit the side, but it wants to keep growing. But again, because we know the mechanics, we know what the force is here simply from the bending of the filament. And then from the videos, it's possible to measure how the length is changing over time. So we know how the growth rate then depends on the force. And that's what's plotted here. And what we see is that the growth is dropping rapidly as a function of the opposing force. It's actually dropping exponentially. And it doesn't agree completely with the theory. The drop is a little faster than it should be, but it seems like we basically understand this kind of force generation. And yeah, let's move it on. Okay, and now we can look at larger numbers of filaments. This is a neat experiment where a bead has a little structure on it that forms a base for actin filaments. It's called an acrosome. And then the actin filaments grow there, and the new subunits add at the end, and they exert force. So in this experiment, it turns out you can hold this piece, this combination of the bead and the acrosome, you can actually hold that in a well-defined orientation, and you can also measure the force on it. And so by doing that, it was possible to measure force as a function of time for this collection of filaments, and it's about 8 filaments that we're seeing here. Okay, so if you look at this curve, you'll see eventually, but it hasn't touched yet. Now here it touches the obstacle, and then the force is generated. But eventually, then you only get like 1.5 piconewtons for all these 8 filaments. So it's kind of disappointing. So the bottom line is that the theory says that we should be able to generate really big forces by polymerization, and the experiments say maybe, but we're not sure exactly how well this works. What's our time?
10 o'clock.
Okay, thank you. Great. Okay. Okay, and here is an even larger collection of filaments. What you're seeing here is it's a bead, and it's attached to a cantilever, which allows us to measure force. And this bead has a cloud of actin growing off the end. So this is thousands and thousands of filaments now. And this, at the end, there's a pipette that's used to grab it to keep it in place. And with this combination, you can, from the bending of this cantilever, you can figure out the force. And then you can simply watch this tail to see how fast it's growing as a function of time. And then again, you can see the velocity as a function of the opposing force. And now, notice that the force up here is given in nanonewtons. And so that's because we have many filaments, and we're basically able to generate forces on the order of nanonewtons per square micron. Okay, now let's look at a whole cell. So this is really getting more biologically relevant. We're going to look at the force generated by a cell as it moves. Okay, and the setup is this. Here is our crawling cell, and this is a cantilever. And by the way, a cantilever is really, it's a narrow rod that bends, and from the amount we know the stiffness and from the extent of the bending, we're able to figure out the forces that were exerted. So in this case, the cell will crawl and bend this cantilever, and that bending is measured via a mirror that's sitting on top of the surface. So this triangle-looking thing that's really just holds a mirror, which allows us to get a handle precisely on how much this cantilever is bent. And this is how it looks from the front. There again is the mirror. And so this has been used to look at cells that are migrating on a substrate. So now the cell has started migrating. Now it's just starting to make contact with the cantilever. You don't see any motion. And force is gradually building up. There's already a force being generated here. We can't see it, but in the apparatus they could see it. But now the cell is moving farther and farther in, and eventually the nuclear mound of the cell moves under the cantilever. Then you get to maximum force point. So now it crawled through, you can see it coming out on the other side. And so during that whole process, it was possible to measure the force exerted by the cell. Okay? And this is what it looks like. So first, the big picture is here. We're just making contact at this point, and you can see the force is like a nanonewton there. Okay, then it drops again to 0, and then eventually it climbs up to a much higher value, like 30 nanonewtons, and that's what's happening when the body of the cell is crawling under. So the basic finding is that initially just a small part of the cell is making contact with the cantilever, and we're not generating huge forces. But later on, we get forces on the order of tens of nanonewtons. Okay, so now let's look at a different question. I was talking before about how there is no net force on the cell, and so it's nice to see that balance of forces, and we can see that by measuring the distribution of forces that the cell is exerting on the substrate. And this is how we accomplish this experiment. So here's a cell with a nucleus, and there's an actin network on the substrate, and it's adhering to the substrate at various places called focal adhesions. And the ingenious part of the method is that the deformation of the substrate is viewed by putting fluorescent beads into it. So here are fluorescent beads all over the place. And so the experiment is done by first taking a video of the substrate without the cell, and then you film the substrate again as the cell moves through it, and then you see that the beads move. And from the motions of the beads, together with what we know about the elasticity of the substrate, we're able to calculate their corresponding forces. Okay. So here's an example of this. This is a cardiac myocyte. So here is the cell, kind of outlined there. And there are various points where the forces are evaluated. And the myocyte is it's one of our muscle cells. This is one in the heart. And so what you see on the right are plots of displacement of the beads on the one hand, that's the yellow, and on the other hand, the forces that are exerted on the substrate. And the scale bars are here. And what you can see is that here it's pulling in this direction on the substrate. Here it's pulling in that direction, and here it's pulling down. And the forces that are exerted on the cell by the substrate are the exact opposite of those. So, for example, here the substrate is actually, it's pulling out on the cell, and here it's pulling out on the cell, and here it's pulling out. And so we have these outwards directed forces, but the forces are vectors. And if you add them as vectors, then it turns out that they basically cancel out. So the cell is sitting there just trying to distort the substrate, but not really moving much as a result. And this is a very common situation in biology. Now, this situation had a particular twist that this cell was actually beating, kind of like our heart. Not that surprising, it's from the heart, but it's exerting strong forces, you can see up to 500 nanonewtons, but then after like a second it stops, and then after another second it starts again. So that cell is kind of like a very tiny piece of a beating heart. The other neat thing about this experiment was that the extent of the force generation depended on the stiffness of the substrate. So the Young's modulus here, that's a way of measuring the stiffness of a material. And they did this experiment with substrates of varying stiffnesses. And what you can see is that there is a huge variation in the extent of the forces that are generated. Here, they're very small, maybe 50 nanonewtons, but here at this other end, they're over 1,000 nanonewtons. So that means that this cell is very clever, because this cell is able to figure out how stiff the substrate is. And it knows that the stiffer the substrate is, the harder it needs to work to do its job. Okay, and so that's the force distribution. Let's talk a little bit about measuring the stress. And to understand stress, just imagine a one-dimensional layer of cells or molecules with a varying displacement. And so let's say they're cells, okay? Now, if we look at this cell here, you see that it's being stretched on either side. So that cell is going to be experiencing a high stress. But on the other hand, because it's very symmetrically placed relative to its neighbors, it's not actually going to be experiencing a force. So the force is 0 at that point. And on the other hand, this cell would be experiencing a large force because it's asymmetrically placed relative to its neighbors. But at that point, the stress is small. And so there's a relationship between force and stress. I don't want to explore this quantitatively. I just want to convince you that if we know the distribution of force, we can also get the distribution of stress. And that's what's studied in these experiments. This is a layer of migrating kidney cells, again, on an elastic substrate containing fluorescent beads. And here is a before and after experiment. So first, the red is the positions of the beads without the forces from the cells. And then the green here is their positions after the cells were put down. And from these motions, it's possible to measure the forces. And so they call it traction here, but these are forces here. And here we get the stress. Okay, and the convention is that a positive stress is tensile, so that means like we're stretching a rope. It's that kind of stress. And so we're seeing concentrations of tensile stress in various places around the cell. But it would have been very hard for us with our naked eye to see that in these pictures. So, but this is something that cells tend to do. They generate tensile stress, and this shows us very nicely how that stress is focused in certain parts of the cell. Okay, and finally, instead of using beads, you can use micropillars, and you put a cell down on pillars like this, and then just look at the pillars and see how much they're bent. And that tells you very directly the force at every point where you're interacting with the micropillar. And in this experiment, they looked at varying degrees of cell spreading, and they found that the more they made cells spread, the larger the average for force, the average force per post ended up being. Okay, what's my time? Okay, there's another way of getting at forces at a greater level of detail, which is using molecular force sensors. And this method, it's called FRET, or first-year resonance energy transfer. It exploits the fact that sometimes if we have a fluorescent molecule, but it's placed close to the emission center, which is called the donor, is placed close to another part of the molecule, which is called, sorry, let's get that back, that proximity of the donor to the acceptor will affect the fluorescence. And so the fluorescence of this green molecule is different in this situation from in this situation. And here, we actually, in the situation of low force, it's compressed and not stretched. And in this case, we don't see the emission from the donor molecule, but when the molecule is stretched, then we see the emission because this acceptor is no longer interfering with it. And this allows us to plot out force on a scale, or on a very fine scale. Now, this experiment it's slightly confusing because what's measured red is actually what's green over here. Okay, so the tension is being measured by doing this fret approach, and that's shown here. And then at the same time, the distribution of the molecule paxillin is being plotted. And what you see here is that the two coincide very closely. And so this tells us that probably the paxillin is part of the force generating machinery. And this is something that we would not have known without these kinds of experiments. So this is a very active field now, designing molecules whose fluorescence depends on their extent of stretching. Okay, another way you can get at stress is just to break something. Okay, if two people were pulling on a string and somebody all of a sudden cut that string, you know what would happen? They would both fall backwards. Well, this is the same principle here. We take some entity where we want to measure the stress, and here it turns out to be what's called a stress fiber. That's a collection of actin filaments interacting with myosin. And here is the target of a laser that blasted the stress fiber. And you can see what happened later is it got broken and the ends moved apart. And initially, the rate of motion is determined by the viscosity of the cell. And later on, you can see that the ends have moved still farther apart, and eventually they settle into an equilibrium configuration that's determined by the elastic properties of the medium. Okay, let's maximize me again. And let's talk about osmotic pressure. So with osmotic pressure, we have a cell, and then we have various osmolyte molecules on the inside, and they exert an outwards pressure on the membrane or the membrane in the cell wall. Sometimes there's both a membrane and a cell wall on the outside, which allows it to support a much greater pressure. And so we'll call this pi. And the formula for pi is that it's equal to KT again, times the concentration on the inside minus the concentration on the outside. Now what I've drawn here is the concentration on the outside is 0. Okay, so let's say that we'd like to measure this Turner pressure. Then one way to do it would be to start adding osmolytes on the outside. Okay. And at some point, when the concentration of osmolytes on the outside becomes equal to the concentration on the inside, then the cell wall will start to shrink. So as we increase this concentration on the outside, eventually there'll be some critical point, or like a cusp, where it starts to shrink, and it might even buckle, it might lose its basic spherical shape. And this has been the approach that's been generally used in the past to measure the osmotic pressure. You just see how much do I need to add on the outside to balance out what's on the inside. Okay, and let's go back to the PowerPoint now. And next slide. And this is an example of such a measurement. Here, the external osmotic shock refers to the osmolytes on the outside. You can see that there's a gradual drop, and then all of a sudden there's a cusp. And that cusp is the concentration that was required to cancel off the pressure from the inside. And so that should correspond to the Turner pressure. And here is an example of measuring this in budding yeast, where the number using this method came out to be about half a megapascal. Okay, so that sounds really good. But what can happen is that cells are really very smart. And so one thing that can happen is that when the concentration on the outside, oh, let's go back to B for a second. When the concentration on the outside is increased, that then the concentration on the inside also gets increased. The cell fights back. It wants to maintain a certain relative pressure between the inside and the outside. So that means that you can keep adding and adding and adding, and you never actually get the cell wall to collapse. And eventually you do, but the fact is that this can lead to an overestimate of the turgor pressure. So let's go back to the slides, and next slide. And so recently a better method has been developed based on nano-indentation. And so if you look over here, here's your cell, and now there's a nano-indenter that pushes into the cell wall. And a very nice physics analysis showed that the corresponding spring constant that you measured as you feel the cell pushing back on this nano-indenter, that spring constant is very closely related to the turgor pressure. In fact, the turgor pressure is just that spring constant times this geometric factor. And so this is the result, spring constant measured as a function of indentation. Here it reaches a plateau, and from that plateau value, it was found that the turgor pressure is actually only 0.2 megapascals, whereas the previous method gave like 0.5 megapascals. So on the basis of that, we are really evaluating a lot of our theories that for probably 10 years, people have been going around with a very exaggerated estimate of the turgor pressure in yeast. Okay, how's my time now?
10 minutes.
Okay, great. We'll talk about this briefly then, about mechanosensitive membrane channels. Almost all kinds of cells contain some kinds of mechanosensitive membrane channels, bacteria, for example, but also pollens. And that's something we're especially interested in this center, because we're combining plants and mammal cells in one center. And in pollens, the way that pollen germination works is that a pollen settles down on what's called the stigma, and that pollen is actually dried out. Then it gets rehydrated, water moves into it, and eventually it forms a projection, and that's what accomplishes the job of the pollen. Now, it was discovered some years ago that pollens have mechanosensitive channels, and experiments were done interfering with those. And it was found that if you made the channels inactive or deleted them, that then the pollen cells tended to explode. And on the other hand, if you made the channels always open, that then the cells never expanded in the way they needed to. So these channels are very important for the function of pollination and also in many other processes. And it seems that they are, each channel has a certain tension at which point it becomes activated. So it's basically a safety valve is the idea. So what you're seeing here is that now there is tension on the membrane and the osmolytes leak out, and then that reduces the tension. And what's very important here is that there's a relationship between the pressure and the tension in the membrane. And they're related simply by a factor involving the radius. So it means that the more osmotic pressure you have, the more tension, and vice versa. And this is what's used to measure the gating values of the tension for the channels. And so this is the kind of experiment that's done. You take a cell, and you suck on it with a suction pipette, and that changes the tension in the membrane. We saw before that when you change the pressure, that changes the tension, and that's what's being done here. Now, if you look very closely at these pictures, on the one on the left, there is basically zero pressure here, and this is the membrane that you're seeing. It's very faint here, but now you're seeing the suctional pressure is larger here, and now you're seeing that it curves out. Okay, and so you know the pressure difference across here, and you also know the curvature, and that allows us to calculate the change in the tension. And so, and you measure the openness of the channels by the extent to which they conduct ions. And so the kind of thing that you can learn then is that for a given channel or a given type of channel, as a function of tension, that it's either open or closed. So this particular type of channel opens up at about 5 or 10 dynes per centimeter. And on the other hand, here's another type of channel that opens up at roughly twice that tension. So you have a whole host of channels in the cell that are opening up at different values of the tension. And we're only starting to understand how those help in regulating the properties of the cell. Okay, so that's really what I have to say. We've seen some kinds of really nice measurements of forces, but it's all very limited at this point, and the technology is really growing, and we expect to see much better measurements over the next 5 or 10 years. And in particular, what we really like to understand is the distribution of stress inside cells, and we're only getting started on that. So I'll call it quits there, and then I'll be happy to take any further questions. And can you then maximize me, Jim? Well, let me repeat. I'll repeat the question. Somebody, can we stop that?
Is there another laptop maybe?
Okay. I think that's better. Yeah. Okay. So the question was, in the nano-indentation experiments, is it possible to do this kind of experiment on animal cells? And unfortunately not, because it requires a cell wall. The geometry just doesn't work out right if you only have a membrane. So this only works for walled cells. So it's going to be good for yeast. It's going to be good for bacteria. It's going to be good for plant cells, but unfortunately not for mammal cells. All right. Thanks a lot.